Everybody, this is Jason from STEM Nerds. Thank you so much for being here. In today's video, we are going to be talking about, well, what is genetics? What is the study of genetics? And we're going to start out with trying to understand some important terms that are going to allow us to study and understand genetics a lot more easily in future videos. So genetics is the study of how genes are passed down from one generation to the next. In other words, it's the study of heredity, how parental genes are passed down to offspring. Now, a gene is a functional unit of the genome. It codes for a protein, and our genome, the human genome, is made up of thousands upon thousands of genes that are spread out amongst 46 different chromosomes. How many genes, you ask? Well, scientists currently estimate that there are somewhere between 20 and 23,000 genes in the human genome. Genetics is a very useful field for understanding these genes and how they're passed down from one generation to the next, and it helps us make predictions as to what offspring are going to look like, and that's part of what we're going to be doing in future videos. Now, it's important to understand that these genes, as I've already mentioned, are housed on chromosomes, and chromosomes are made up of DNA. This picture that you're seeing here is a section of DNA. It is a double helix. It's got a backbone, and then it's got base pairs, nitrogenous bases, holding the two strands together. And when it comes to chromosomes found in the human genome, we like to organize them based on size. And here you can see that there are 23 sets of chromosomes in each person. This number one set is the largest of the chromosomes, and then you go all the way down, and the first 220 sets are known as autosomes. That last set is either going to be an X and a Y or XX. Those are known as the sex chromosomes. What's pretty interesting is what you're looking at here is DNA fingerprinting, and this allows us to differentiate organisms or different people based on the structure, the sequence of their genetic material. One of the ways that we study heredity, or the way genes are passed down from parent to offspring, is using Punnett squares, something that we will tackle in a future video, but this is just an example of one where if we know the characteristics that we're looking for, and we know the genes that are contributing to those characteristics, we can figure out or calculate the probabilities that those characteristics will be passed down from parent to offspring. Now, something that's really important for us to understand is that the DNA has a specific structure, and that leads to its function. The DNA is all of the genetic material found inside of a cell. The DNA, as I've already mentioned, is a double-stranded double helix. Now here, we're seeing the double helix on the far left-hand side. We can see that it's double-stranded in the picture in the middle. You're going to notice that on the left-hand side, the gray portion, that is the backbone of the DNA. And those are held together by covalent bonds that are very, very strong. In the center, you're going to see what are known as nitrogenous bases. And those are the letters represented by A, T, C, and G. And you can see here on the left-hand side what those letters stand for. You've got guanine, that's your G, cytosine is C, thymine is T, and adenine is A. And when you're structuring DNA and you have the two strands, for every A, across from that is going to be a T. For every C, there will be a G. Now, what's holding the nitrogenous bases together in the center are hydrogen bonds. Now, individually, they're usually very weak, but when we start putting lots of them together, they can be quite strong, and that's what's holding the two strands of DNA together. You're going to notice that gray background also has some important structures. One of them is a sugar, and the other one is a phosphate group. In a future video, we're going to go into much greater detail as to why all this structure matters and how it contributes to the structure of DNA. The DNA that we have inside of our cells is eventually transcribed or copied into RNA. It's like taking the DNA, which is a cookbook. It's got all the recipes for every protein that you would ever need to make inside of a cell. But when we need to make that protein, we take the DNA and we transcribe it or copy it into RNA, which is a single recipe from that cookbook, and that's on the left-hand side. And so what we have to understand at this point is that DNA and RNA have some important differences, one of which we've already said the DNA is a double-stranded helix. The RNA is usually going to be, at least in human beings, it's going to be single-stranded. Nitrogenous bases are a little bit different for the RNA. You're going to notice that there are no T's in RNA, no thymine. Instead, we have a different nitrogenous base called uracil, and we usually designate that with the letter U. And of course, because RNA is usually just a single gene, it's going to be much smaller than the DNA, which houses all of the genes for an organism. One other important difference is the sugar itself in the background, not shown in this picture, but we will talk about it in a future video. The sugar itself is different, and that's one of the main reasons for the different name. RNA stands for ribonucleic acid, DNA, deoxyribonucleic acid. And so in DNA, where the sugar exists, there is a missing oxygen. Again, something that we'll cover more in a future video. When we're studying DNA, a lot of times scientists focus on what are known as exomes or exons. These are the parts of the DNA that are expressed. Turns out that DNA has a lot of parts that are not expressed to make proteins, and those are called introns. And so traditionally, when scientists have studied DNA and the sequence information to figure out what parts of DNA really matter, what contributes to the differences that we see between human beings, what contributes to the differences that we see between human beings and other animals, it's the exons or the exome that has primarily been studied. But scientists are quickly realizing that they need to look at these introns, and even though they don't code for proteins, they may hold important information as to why we see differences between human beings and different organisms. One of the really interesting areas of study right now in genetics is the microbiome. It's the microbes that associate themselves with human beings. These microbes provide all kinds of benefits. They make certain vitamins for us. They protect us from pathogens. They help our immune system grow stronger. And we're really trying to figure out all these microbes and what they do. And it turns out they line the surfaces of our body, both the external surfaces like the skin and the internal surfaces like mucous membranes. And so microbes can be found in the upper respiratory system. They can be found in the small and large intestine, and they can be found in the genitourinary system. And these microbes provide benefits. It's one of the things that we've learned is that we can't culture and grow all the microbes that exist on or in the human body. And so what we do is we try to study their genetics. From there, we try to figure out what important roles the microbes have associated with the human body. Now, because we can't grow all of these microbes in a lab, we study their genetic information, and then we can make inferences based on the genetic information that is present, what types of microbes are growing on the human body, and the communities of microbes that we have associated with those mucous membranes. The genome is just packed full of information, but here's the cool thing. DNA doesn't just exist inside of our nucleus. It also exists in the mitochondria that has its own set of genetic information, and that is also a hot area of study. And we can use the information that we learn about the DNA in the mitochondria versus the DNA in the nucleus to determine how different people groups are related to one another. All right, everyone, so that is just a quick introduction to genetics, the study of genetics. In future videos, we're going to be doing a lot more when it comes to studying heredity, how genes are passed down from one generation to the next, and we'll be looking at applications of genetics and looking at biotechnology. As always, thank you so much for being here. Have a great week.